Swayam Prabha. Digital India. Educated India.
Hello everyone. So I'm Vaishnavi. I'm a PhD student in Professor Gohan's lab, and today I'm discussing my PhD work, what I've been doing all 5 years. It's been a long time, and how I have used some of the synthetic biology tools for my PhD. I also like to emphasize on those things so that you can get to see the real application of what we have been taught so far by Professor. So as an introduction, so basically my work is lignocellulosic biomass. So what are they? So they plant waste materials which are abundantly available. So as we there is a definition of fossil feedstocks, we have natives. These are one of the wonderful source of resources that can be exploited here. So when you look at the cellulosic biomass, they everywhere waste residues in the form of agricultural waste, forest residues, and so on. So what they do is they just exploit the cellulose and cellulose part of it. They just convert the cellulose to glucose, and they do fermentation for production of variable products. But mostly the hemicellulose part, which is containing xylose, part, which is the most underutilized part. So they just burn the part of it and then just use it for production of energy and things. So instead, lignin can be valorized to produce some bioproducts. And yeah, so this is the scheme of a biorefinery where we have this in our lab. Typically, we use these two biomass like rice straw and corn cob, the waste residues, and we use enzymatic hydrolysis for the conversion of cellulose to glucose and produce products like lactate and so on. And we also have this hemicellulose fraction, which is mostly xylose rich, which is also underutilized because bacteria do not take up xylose like they take up glucose. And yeah, so we can able to find which can take up xylose. It will be like using a underutilized part, and also this lignin, just the least utilized. We can convert them so to products. So we have valorization, which I will be mostly focusing on to this complex lignin molecule, which have a lot of aromatic compounds. We have to undergo a process called depolymerization, where we have to break them into small monomeric lignin-derived molecules so that it's easy for the bacteria to take up, and then it will do bioconversion with the pathways available in its metabolism for the production of value-added chemicals. So what I have done is I have chosen some of the lignin-derived models like ferulate, cumarate, caseate, vanillate, 4-hydroxybenzate, and I have used strain known as Pseudomonas taiwanensis VLB120. As we have heard a lot about Pseudomonas, it's a fantastic strain. It can take up a lot of unusual substrates, and it has high tolerance. And the one major advantage of this strain is it can also take up xylose without need for any engineering pathways for xylose. That's fantastic. So it can take up sugars, things, everything. So that shows the strain. So this is the aromatic degradation pathway. Generally, it's in other Pseudomonas strain. It does also caseate. Those are very difficult. It can only take 4-hydroxybenzate and vanillate. So what we did was we took genes from a pathogenic Pseudomonas strain, which is called Pseudomonas cotida S12. That's very pathogenic. So we took strains from that and we genetically integrated that strain, the genes, these three genes, the which was responsible for the of these complex molecules, and we see we were able to achieve growth of substrates. So it was able to take only 2 substrates, and in the engineering step, it was able to substrates. So this was very nice, and then we so we used a strategy which we are which is known as adaptive evolution. A subject strain the stress conditions, and then we increase the tolerance towards these toxic substrates, and we carried out a for 2 days for several generations, and we got strains which could grow better at concentrations of these toxic substrates. Like if you can see the non-involved strain which had a longer life phase, didn't have much life phase, and also on these substrates, and also there was at least 2 to 3-fold enhancement in the specific growth rates, which was considered to be the fitness thing. So the aliens very nice to update strains with enhanced phenotypes, and also gene sequencing analysis to understand what kind of mutations happened in the evolved strain that led to enhanced phenotype, so that they can be used as targets for development of platform strains with enhanced tolerance. So these are some of the interesting targets. There were a lot of other mutations as well, but we found these ones more relevant. So we just had some of them as targets. Acetate importer, which was responsible for the transport of acetate molecules into the cell. So there was mutation, which was SNP mutations like SNPs and indels, and there was also mutation regions of regulators, HGD, and also mutations in degradation of phenyl acetate. So they have tolerance, and then we also some platform strains, some expression of some of these genes, and yeah, like you could see here, the VS1 is the non-evolved strain, and VS2 was the evolved strain, and the rest were the reverse engineered strain, and the reverse engineered strain showed intermediate between these two, because there could be the cumulative effect in the final strain. These can be also potential outcomes because the phenotype is better compared to the non-evolved strain. So we took strain, some platform strains, production of industrially important biosurfactant, which is known as rhamnolipids. So basically, this biosurfactant has long potential application in various industries like industry, medicine, oil, cosmetics, and so on. And market, it's also so we can produce some detergents out of this from our waste. It's really nice. So what we did was we took the lipid production genes, and then we just genomic integration transport system, which we learned, and we were able to achieve with just these substrates. So yeah, good titers, maximum titers of which was in conventional substrate, and we were able to strain this metabolic engineered strain for production of first concept, and then the real is to some the lignin residues stuff. So we did in order to depolymerize this complex molecule, we did a biodegradation experiment to break down this complex molecule and see the metabolically engineered strain. But you see, like too many compounds. When we do biodegradation, it took a lot of time, like at least 4 to 5 days, and the yield of individual monomers were very less. So, like if you could see, these were the compounds that were the degradation of like the controls, these compounds are not like the control is the one which is untreated. But if you could see the treated sample, lot of these compounds, and some of our reactors here, like cumulative acid base, or really. So we had to go for degraders which are chemicals. processes, they are much faster, they can give higher selectivity towards particular. So, picture, we found that like pyrolysis, for example, gives high molecular weight lignin, which are difficult for the organisms to take up. So what we did was we took a chemical process known as mild base catalysis, where we were able to achieve cumarate as the main degradation product with higher yields. So we had this concope with the hydrolysate of the batch enzymatic hydrolysis, which would be rich in lignin. So after the cellulose part is converted to glucose and it's exploited, after fermentation, we will have a biomass which is lignin rich. So we took that and did a chemical mild base catalysis at 120 degrees, and then we analyzed the products. It was mostly cumarate, but yeah, if you could see the products here, it was mostly cumarate with very minimal levels of other substrates. So we deal with processes, it's always better, higher yields, and so we got a really nice profile just having cumarate. So we did this. We took this as substrate for the produced rapid. So it's also very good, as you could see. The metabolically engineered strain was able to just grow on this depolymerized lignin as substrate. And yeah, so right now we are focusing on using this depolymerized lignin for our production experiments. And also we are looking at utilizing the xylose rich fraction because this strain can take up xylose. We can also utilize that part. So it's like toxic biomass. Yeah, so that's the ultimate aim of my project. Yeah, that's it. Thank you. Basically, they are biosurfactants. They are used in a detergent industry. Also, they have some pharmaceutical applications as well. Yeah, and there is a big plant now being set up for just rhamnolipid production itself. I think it's by Unilever. Yeah, yeah, they are also used for bioremediation. They can remediate spills and so on. They are also applied to that as well. Yeah. Thank you.
So I'm working on the design of genetic circuits under the guidance of Dr. Karthik Raman and Professor Raghunathan Ragasan. A bit on the view of synthetic biology that I have in mind in my work. I'll talk a little bit about that. So the way I see synthetic biology is it can be distilled into three abstraction perspectives. One is the biological perspective that we have been talking about in this course. Then there is the network perspective, and then circuit behavior perspective. So in the biological perspective, we would actually talk about and think about all the processes involved in the synthetically implementing the circuit. And then there is the network perspective, where we think of the genes and their interactions as just nodes and edges. And then we try to look at what kind of interactions bring about what kind of profiles in the concentration of the gene products. And then that is a way to design as a graphical problem. And circuit behavior is when we look at the circuit as an inverter or a NOT gate, logic gate, or oscillators, and so on. So I work mostly with the network and circuit behavior perspective, and not so much with the biological perspective. But then, as we realize in this workshop, at least, that it cannot be completely independent. Probably it would benefit to get in elements from all the perspectives while we are working with along any of these arms. There's one more important point that I want to make over here is, in my view, circuit design and circuit implementations are two separate tasks, where computationally designing a circuit need not get into too much into the details of how to implement the circuit. So clear division between design and implementation. So in my firstly, what I'm trying to do is to come up with a framework in which I can which I can use to computationally come up with the blueprints of the circuit that a biologist can take to the wet lab and design actually using biological components. So for this, the genes are the nodes, the interactions which might be activation or repression, they can be the positive or negative edges between those nodes. And then the dynamics of the system is modeled using ODEs. And typically for computational studies of genetic circuits, we use Hill kinetics. And using these ODEs, I would try to design circuits that have functionalities as listed here. And of course, the list keeps increasing every month. And then I would simulate all the ODE models. Typically, that is what is done. And I get the concentration versus time profile. And when I simulate for large enough number of circuits, I get an understanding of what kind of G's bring about what kind of functions. So there is some work in this area where they have shown computationally, purely computationally, that what kind of topologies are needed to get adaptation in circuits. And in my work, what I'm trying to do is once I have these simulation data, I form this topology function map, and using this simulated data, I train ML models and then use some generative methods to come up with novel circuit designs, which tells me not just the topology, but also the parameters, so that when somebody wants to take this design to the lab, they can map with the map components that show similar parameters and then put them together and check whether this design is actually it can be implemented and the profile that we are predicting can actually be achieved in the lab. So this is the first objective that I'm working on. Now, the question, we are always in doubt whether a circuit which is in a blueprint form, whether it will work out. So there are some methods to check the reliability of the designs. So one of them is the parameter dependent effects. So when I write the ODEs, I have Hill kinetics, so I will have a lot of parameters like the protein degradation rates, the molecular degradation rates, the Hill coefficient and all that. So how does a given circuit behave with changes in these parameters? So there is this mathematical method called bifurcation, which helps us understand which regime of the parameters will give us the desired output and which ones are to be avoided. So here I've given an example of a toggle switch driven by an oscillator, and I simulated this model. They coupled in a linear fashion, and then a bifurcation analysis reveals which part of the parameter space is going to make sure that the operation of the two circuits is such that the first circuit is driving the second. So this bifurcation analysis I have done for the coupled circuit shown before, and also I'm currently extending it to adaptive circuits so that we can see what kind of parameter changes make a circuit lose its capacity to adapt. And then the idea is if we have a circuit like a toggle switch driven by an oscillator, can we equate with the biological scenario where the circadian clock is driving the expression of a gene? And the long-term vision is to see how this can help in personalized chronotherapeutics, where we can probably have a synthetic circuit which tells us the circadian status of a patient and then tells us exactly which time of the day therapy should be administered, because there is some evidence that the time of day matters in the therapy of certain cancers like colorectal cancer. And so chronotherapeutics is one area where we could probably find an application of such circuits that are working with probably in the long run. Another problem that we find is retroactivity, which can be equated with the problem like the couple tanks, where we have two tanks, they have some inflow coming and there are outlets and they are coupled by a passage which they share. So now the task is to maintain the same level of water in the two tanks, Then there is this method called feedback linearization, which is a nonlinear control design method, which tells us how to design the inputs U1 and U2 such that we can maintain the same level of water in both the tanks, given this coupled scenario. So my idea of handling retroactivity in generic circuits is to think of retroactivity uh, as a similar effect that comes into play when we bring together two different circuits. So, so we have actually discussed in this workshop how retroactivity is a big challenge in synthetic biology, where we put together well-characterized parts, but then when combined, they lose their individual functionality, and that's not a very good sign for synthetic biology, because we would want to compose bigger circuits using well-characterized parts. So, in order to skip this, I think there is some work in the literature where typically they consider the downstream circuit as just a node connected to a circuit, and they call this as a canonical downstream node. And this model does not really give a very good idea about how to handle retroactivity computationally. Instead, I have shifted the view to something like this, where we have two circuits with their individual inputs, and then there are their individual outputs, and the red arrow indicates an interaction between the two, which is not part of the design. So using feedback linearization, we can design the kind of inputs that we need to put to the individual circuits so that they behave as if they are not coupled at all. But then there can also be scenarios where we want the circuits to drive each other, but there might be some interactions which are also non-intended. In that case also, feedback linearization gives us some methods by which we can design the input so that only the intended interactions are happening and the unintended interaction is cancelled out by the way we design the input to the circuits. So this is kind of broadly what I work on, and that's it. Any questions? Okay. So it's multiple outputs systems and single input, single output. Sorry, I did not expand it in my slide. Yeah, so they are actually not orthogonal, but we can design some how to vary the inputs so that they behave as if they are orthogonal. So that is the linear control design method. So it is possible to do theoretically. I'm still kind of working on that. We might design the input to be, let's say, a particular variation, and we would have to put additional circuitry to achieve that kind of an input, and we can check whether it is really cancelling out the unintended interaction. So generally in her work, she has used insulators in between two modules, and probably she has found some constraints, which will ensure that these are cancelled out. Yeah, so the non-interacting, like unintended interaction and intended interaction, this coin was termed by her only in one of her papers.
Sure, please.
Hello everyone. So the topic our team has chosen is this one: bacterial drug delivery system in synchronization with human gut circadian rhythm. So this work was mostly inspired by the work Biomedical is doing in her lab. So you will find some similarities with her presentation that you just saw. So we basically had a different vision for this work, but then when we went looking for the parts and literature on the topic, we ended up with something very different from what we initially anticipated. So this is mostly based on the paper that is on the screen right now. Human gut bacteria are sensitive to melatonin and express endogenous circadian rhythmicity. So this paper basically shows how human gut bacteria, specifically this bacteria, Enterobacter edogenesis, can show some circadian rhythmicity. I will go into more details about this in the next slides. So circadian rhythms are fundamental properties of most eukaryotes, but there is not much evidence of this occurring in most bacterial cells. We know this occur in photosynthetic bacteria, but in other bacteria, there isn't a lot of evidence for this happening. So this paper basically takes out the enterobacterial urogenesis from human gut and tries to see if it shows some circadian rhythm. And as it turns out, the bacteria does show some circadian rhythm, and it is actually related to the melatonin production in the gut. Now, the melatonin production in the gut is basically not in complete sync with the core human circadian clock that is mostly regulated by some of the clock genes. I have forgotten the exact names. This is kind of the gut circadian rhythm is a bit out of phase. If I remember correctly, it's like plus 2 hours or something like that. So the advantage of this bacteria is that it takes the data directly from the gut. So it is perfectly in sync with the gut circadian rhythm, and it also takes some other factors into consideration, like the temperature and stuff. So basically for this bacteria, the melatonin levels are used for entrainment of the oscillation. Another important thing is that this bacteria is, because it lives in the gut, it is kind of personalized for the gut of the person it is sleeping in. That was another problem that we expected to face, because the melatonin levels in the gut of every person will be a bit different, but this bacteria is perfectly adapted to that. So this is just a bit about clock genes that regulate the circadian rhythms in mammals. So it basically, the circadian rhythm comes from the expression of clock genes, which interact in dynamic transcription or translation feedback loop. And the important part is that we really don't have a complete picture of which clock genes are actually completely necessary for the circadian rhythm, because in some experiments they have knocked out some clock genes from rats and they will still show the circadian rhythm. So most probably there is a host of genes that are regulating it, and it is not regulated by one gene or two genes. Another thing this paper goes into is how exactly did this Acetobacter aerogenesis get the circadian rhythm genes? Did it occur by horizontal transfer, or is it an inherent feature of bacteria? So the hypothesis is that it is not an inherent feature of bacteria, because other gut bacteria like E. coli does not show any circadian rhythm or any oscillation based on the levels of melatonin. So they went into some genome sequencing and tried to correlate the genome of the bacteria with the human genome, but nothing notable was found. At least no clear result was found about this. So we are still in the dark about this, how exactly this bacteria acquired these genes. Now I will go into what is the main role of gut circadian rhythm in the human digestion and regulation of the human gut. So recent studies have suggested that microbial signaling plays a critical role in homeostatic maintenance of intestinal function along with the host circadian mechanism. So this has been kind of a research area for a long time, where people have been trying to understand the interaction between the human gut and the human nervous system. But right now we don't have very solid data on this. So this is completely a hypothesis, but this melatonin level and this bacteria is something we have the data on, as shown in this paper. So we have the evidence for the indole hormone melatonin, which present at high levels in the gut, and it in turn induces swarming activity in the clinical isolate of the Enterobacter aerogenesis. So what basically happens is that when the melatonin level in the gut peaks, the Enterobacter aerogenesis shows a swarming activity. It forms clusters, not exactly biofilms, but it forms a kind of clusters of cell. If you want more details, you can go into the paper, and they have a diagram to explain this. We did not have time, so we omitted this diagram. So when the colonies of this Enterobacter aerogenesis was proliferated on semi-solid agar, significantly more rapidly in the presence of it grew significantly more rapidly in the presence of melatonin in a specific dose-dependent fashion, with maximal response coinciding within the physiological range of gut melatonin level. So the important thing to note here is that the maximal response will occur in the maximum gut melatonin level that the bacteria is used to, which is the maximum gut melatonin level of the guy the bacteria grows inside. And this effect was specific for melatonin, as this aerogenesis further spread further in the presence of melatonin than in the presence of equimolar concentration of other things like tryptophan, serotonin, or N-acetyl serotonin. Importantly, serotonin, I think, is a precursor for melatonin. In contrast, other bacteria which are closely related, they did not show any kind of oscillatory or growth behavior when exposed to high melatonin levels. So this paper basically establishes that E. aerogenes bacteria shows a oscillatory behavior in presence of melatonin, and when the melatonin level peaks in the gut, this shows a swarming behavior, forms clusters of cells, and this is not observed in any other bacteria in the gut. So this is where we plan to take the data from when the gut melatonin levels are peaking, and basically get the details about the oscillations in the gut circadian rhythms. So another thing they did in the paper is that they hypothesized that the swarming rhythms might represent the outputs of a circadian clock, and to test this, they transformed the cultures of E. aerogenes to express luciferase, which is a bioluminescence protein. So they basically used bioluminescence to express to basically test their hypothesis. So this also establishes the fact that this bacteria can be easily made to express luciferase or some other bioluminescence protein. One interesting fact to note here is that this actually works only in the range of the human body temperature. So I have discussed a lot about melatonin here, and I think we need to go into why melatonin is the ideal candidate for an application like this. First of all, melatonin reduces oxidative stress in the gut and also reduces inflammation. So there is some research going on the effect of melatonin in the gut and the potential use of melatonin as a medicine for reduction of gut inflammation or for the treatment of specific types of gut cancers. Another interesting thing to note here is that the melatonin level peaks for two different events. It peaks during feeding time, and it also peaks in the event of prolonged starvation. So that is another important thing that they have noted in the paper. So basically, we are using this bacteria to detect the peak melatonin levels in the gut and bring our oscillator in phase with the human circadian rhythm in the gut, not in the entire body, but specifically the gut circadian rhythm. So to sense when the gut circadian rhythm is peaking or when the melatonin level is peaking, we need some form of quorum sensing, because the bacteria is actually forming colonies at that time, and that is the only physically observable behavior that the bacteria shows in when the melatonin levels are high. So we wanted the output for the quorum sensing to be something completely orthogonal, so that it doesn't mess with anything else in the gut and we don't get false positives or false negatives. But we did not have a lot of time to research on whatever happens in the gut. So we went with the thing we were pretty sure was orthogonal to everything in the gut, right? So basically we are using the bioluminescence of this bacteria or to the alter strains of this bacteria, get an idea about when the swarming behavior is happening. So for that, we actually are using a light switch from iGEM team. It was used in 2014 and it was actually repurposed from another paper. So this switch basically is a light responsive gene that activates using far red light. The far red light sensor works as a switch that inactivates the expression of another system that they were using in this, but that is not relevant for us. In the literature, the switch also used two different light waves, deep red for the activation and far red for the immediate and permanent deactivation. But for this project, that has not been used. So it's only to turn on the thing. So this is basically the details about how the entire device works. So basically, in the presence of far red light, the photochrome and the EIF6 complex will transform to its active form, and subsequently it will activate the production of small interference RNAs. And these interference RNAs will bind to the degreening system and trigger the system. So this is basically the switch we are using, but we are not, of course, we are not using the degreening system that has been outlined here. We will be using it for something else. We don't have the data for whatever else we might be using this for, whatever drug we want to be producing. So I have left that part out. Now we come to the potential applications of this. The potential applications of this is purely in the form of chronotherapeutics, like Devamita already underlined, and also for combination therapy with things like melatonin, because this is directly correlated with the melatonin production. And melatonin is actually a very promising candidate for treatments of gut ulcers and gut tumors, and also for diseases like inflammatory bowel diseases, because of its antioxidative effects on tissues, and also because it because of its antioxidative effects on the tissue, and also because it aids in tissue repair. And the literature is already present to point to the fact that melatonin can be a potent medicine for these disorders. So our potential application goes into the realm of using another drug in combination therapy with melatonin. And our main advantage here is that we can use the drug and we can time it correctly, because we have an organism that is perfectly in sync with the human melatonin system, human circadian cycle. And another advantage we have that is that because we are not implementing the oscillator, and the oscillator takes multiple inputs and has evolved naturally, the oscillator is much more accurate than anything we could have implemented by ourselves over the course of one day. Thank you. That's all.
Thank you. Questions, comments? Yes.
Suppose let's say you don't know the melatonin causes the blood-brain barrier. Once you take the blood-brain exposure path, then how is it possible that this will cause later effects of?
I don't think so, because we are not directly producing melatonin. We are just using the melatonin that is produced by the cell, and we are using the switch to produce something else. So what are the side? If any side effects will occur, they will occur. Other drug that we are utilizing based on the melatonin. So what this is basically detecting when the melatonin levels are peaking and bringing something in sync with the circadian clock in the gut. I think melatonin can cross the blood-brain barrier, because I read a few papers which outlined that some of the melatonin in the gut actually originates in the brain. I think that can happen, or that actually happens in the body.
You say that like that using melatonin now.
Not using melatonin system, using melatonin in combination with melatonin. This is synced with the production of melatonin. So what I basically need to do here is that we need to the patient the altered states of the bacteria, and some of the altered states will be the photoluminescent bacteria, and some of the altered states we don't know the ratio. The ratio can only be determined experimentally. Some of the altered states will have the light receptors. So what happens is that when they are they don't have any light. The light receptor is not activated. So yeah, when they're forming community, my hypothesis is that environmentals can detect the lights from the bioluminescent cells. And that is the light detector switch, which in turn kickstarts the production of a drug. That we have no data on.
Okay, so what is the structure of melatonin? Is it a hormone, is a peptide thing, or is a chemical structure? What is the what is chemically melatonin? Yeah, but this is a small molecule, or is a peptide, or how, what is it? We should look for it. Okay, let's check. Yeah, because you know, one possibility that I'm thinking is that perhaps you can engineer probiotic bacteria also that produces melatonin,
That increases
and then well, you would change entirely the circadian cycles or part of the behavior of day-night cycles in
yeah.
Fighting the amount of I don't know.
It is correlated to the sleep cycle. But we are talking about the gut only, so it cannot cross back into the blood from the gut. So all the organs, they kind of the circadian rhythm, apart from the main circadian rhythm of the body that controls the sleep-wake cycle.
right, is a small molecule, right? As in indole containing molecule. And well, okay. It will be interesting just for in other context to see whether you can synthesize this in E. coli, right? And have it secreted, not from the host, not from the human host or the animal
Yes,
host.
maybe
Exactly. Okay, very good. Right. Yeah. Yeah. Right. Yeah. Right.
this paper where they have already identified something that it happens naturally. So we didn't have to put that to other areas.
Well, very interesting project, very well thought, and well, maybe it could work. So, congratulations. Thank you very much. Okay, great, ladies and gentlemen, let's continue with the next one. Who's the next one? You? Okay.
Hello everyone, I'm Dave with Rasa, and for our HMI project, we were looking for a solution to combat lactose intolerance.
Microphone.
Okay. Sorry. So yeah, we were looking for a solution to combat lactose intolerance. The general idea that we came up with was to produce recombinant lactase in the E. coli. That will be a part of the gut microbiome. So basically, you get lactase inside the intestine.
So coming to the problem statement that we've taken. So lactose intolerance is a very prevalent thing that is found around the world. Around 60% of the adult population is affected by lactose intolerance. So lactose intolerance essentially means that there is a decreased ability in the gut microbiome to digest, a decreased ability to digest lactose. And lactose intolerance is basically caused due to the lack of the enzyme lactase, which is involved in the breakdown of lactose into glucose and galactose. So coming to the function of lactase.
So as mentioned earlier, it helps in breaking down the lactose into glucose and galactose. And it does this by breaking the beta glycosidic bond in the lactose. This is absorbed through the intestinal walls and is taken to the bloodstream for further.
So, is an outline to the solution that we propose. We propose to engineer the E. coli missile 1917 strains. Why we specifically chose these strains was because they've been shown to be biotic, which means that when they are introduced in the gut microbiome, they would be safe and maybe even improve the gut microbiome overall. The other major challenge that we wish to tackle was that lactase typically in the is produced only mostly in the duodenum and the small intestine. So we would like to specifically have the E. coli colonize these regions of the large intestine and the small intestine. Yeah, and further we plan to use the PTRH1 plasmid. Here we have made a kind of outline of the gene sequence that we wish to insert in the E. coli. The interesting part that we would like to point to is that it's going to be an inducible promoter. It's going to be regulated by an inducible promoter, and the induction will be done by lactose itself. So when the lactose concentration in the small intestine goes up, then the E. coli will produce the lactase using the LCT gene, which is the gene that produces human lactase. These are the challenges that we feel we will encounter on the way. So then the E. coli chassis should be effectively able to colonize the gut. Also, now the lactase will be produced inside the E. coli, which thus we would require the E. coli to get the lactase out of itself. Hence, I believe that certain amount of membrane engineering would also be required. And the production of lactase would also require certain raw materials. Further on, this is only a very basic outline of the idea that we came up with over, say, past 16 hours or something. So yeah, the regulation and the expression challenges regarding that remain.
Thank you. Okay, thank you. Okay, questions, comments? Okay, so I want to make a few comments. Well, first of all, thank you very much. I think it's a super nice idea. I wonder whether anyone else has figured out already something similar.
We took to train trials which that would manipulate the lac operon itself in and aid lactose intolerance. But our idea was to produce lactase itself inside the gut microbiome instead of having, say, the human system produce it.
Okay, so there are various things that are interesting. Some ideas are really good. Oh, well, okay, so this is the thing, okay. Okay, well, there's a little detail is that silase cannot be induced by lactose. Silase responds to benzoate, so they'll be careful about that. So that's a little detail. Okay, the other question is whether it would be a good idea to secrete the enzyme rather than produce the enzyme intracellularly. You know what I mean? So instead of having a bacterium that has the enzyme inside and you have to import the lactose and everything, how about either displaying the protein on the surface of the bacteria and or to secrete the protein altogether to the external medium? That could help.
Yes. Basically secreted.
Okay, however, the enzyme is multimeric, right? So it's so how many monomers have there? Like 2 or 3, 4, right? Yeah. Well, you know, sometimes secretion of multi-domain proteins is a problem. Yeah, it's a problem. But in any case, it's not impossible. So that's something that perhaps could.
Even displaying it on the cells.
Yeah, it's a problem. Do you know where the active center is in the enzyme? In the little pockets here? Okay. Or they are somewhere around. You don't know. Okay. Yeah. Yeah. And the other detail is that you propose to use the initial strain that are you familiar at all with this initial strain? No? Okay. You know what the origin of the initial strain is? Okay. Okay. You know why it's called 1917? Exactly. Okay, so the strain was isolated during the First World War because one British, no, I'm not sure whether it was British or it was German. I mean, one of the contendants in the war, in the trenches, obviously there was dysentery and there was diarrhea and this was all types of horrible things. But then one of the local MDs saw that one soldier never ever got intestinal problems. And then he said, well, maybe it's because it has a different strain, so I isolated this strain, and it turned out to be one that is very protective. So this strain indeed is a probiotic, can be bought in pharmacies in Germany. I have bought it myself for various purposes, and it has been generally considered as safe. However, recently, it has been found that it secretes also some mutagenic products, and whether or not it's completely safe is now a question mark. But in any case, conceptually, it's a very nice thing. Obviously, you have other bacteria in the gut, and maybe you can engineer something similar in other bacteria in the gut as well. Yeah, you can make a nice engineering. Okay, well, thank you. I think that it's very good, and who knows, this may even work and result in a commercial product. Okay, well, thank you very much. Okay, can you put your names there? Yeah. Because then what you do is that you feed the biomass, what you want is to destroy the lactose, not to, get the E. coli overgrowing the rest of the microbiome, right? Okay, is that clear? Yeah. Yeah, but obviously you have to consider all these different scenarios. And even if you make the best prediction, at some point you have to give that to a patient or to someone and then see what happens. You never know, maybe you have a side reaction or something. So this is something that you have to consider. Okay, next team. Can we tag peptidase bacterial proteins in order to secrete? Well, I think that this is a bit connected to the discussion. So obviously, depending on the protein that you want to secrete, you have to consider various mechanisms. So I don't think that protein can be secreted as such simply by putting a single acynyl peptide, but it is possible to consider other secretion systems. So that's not impossible, and the question is very good. So it will be something for further discussion and so on and so on. Okay, 3 people in this team.
Good afternoon, everyone. I'm Pooja and this is Adi. And the problem statement we're going to tackle is using selenomelanin as an effective radiation shield. Yeah, so why exactly do we need this? So the thing is, when you go to space, the radiation levels are extreme. So an astronaut aboard a space shuttle to the ISS, which is a duration of 6 months, receive about 50 to 2000 MHz of radiation, which is basically in the same range as the survivors of the atomic bomb explosions in Hiroshima, Nagasaki, and long-time workers at nuclear factories. And even astronauts, once they're aboard the ISS, they have a daily dosage of huge amounts of radiation. So this obviously causes various health problems in these astronauts. So we would like to propose a solution so that they can have safer journeys. So for this, we're going to recombine E. coli to produce selenomelanin, which will then be used in space suits and space shuttles, etc. So for this, the chassis we're going to use is E. coli because it is very simple to recombine. It's highly flexible. So the thing is with E. coli, it does not naturally produce melanin with its existing pathways. So we will need to encode a tyrosinase production pathway into the E. coli, which this then expresses on the surface. So
okay, so now as Pooja mentioned, we're going to be expressing this tyrosinase that we're producing onto the cell surface. Now, okay, so before we get to that, I just sort of want to point out the current existing pathway for one of the types of naturally occurring melanin that's possible. We're going to be modifying this pathway of the production of thiomelanin. So currently what happens is the amino acid tyrosine sort of gets converted to DOPA in the presence of tyrosinase, that's dihydroxyphenylalanine.
And again, in the presence of tyrosinase, it gets converted to dopaquinone. And in the presence of cysteine, it sort of gets, converted into other products, which upon oxidation form pheomelanin. Now, our sort of aim here is to convert the pheomelanin, I'll show you this later, we're going to be modifying it to produce a new sort of melanin called selenomelanin. Now, this selenomelanin is going to be manufactured in sort of like a very similar pathway, with the exception being that instead of cysteine that's acting here, we're going to be using selenocysteine. So as Pooja mentioned before, we're going to be doing this on the cell surface. Going to be expressing the tyrosinase on the cell surface, which is then going to undergo this entire synthesis of the melanin. That's mainly because that reduces the cytotoxicity of the cell. And it also improves the reaction efficiency in the production of the new form of melanin that we're calling selenomelanin. So, right, so basically, we're going to be taking selenocysteine and tyrosin, and in the presence of tyrosinase and oxidative polymerization, it's going to form selenomelanin. Now, this is the only difference in pheomelanin and selenomelanin. This was, in pheomelanin's case, this is a sulfur, which has then sort of been changed to selenium. Now, the reason why we think this is going to be a better, why it's going to be more effective against radiation is because the absorption of radiation is proportional to the atomic number of an element raised to the 4th power. So sulfur is 16, and as you guys know, selenium is 34. So essentially, the absorption coefficient of selenium is 20 times greater than what you can see in a normal, naturally occurring pheomelanin. Right, so the way we're going to be doing this is through a plasma, we're engineering the E. coli cell. So we're taking a plasmid, P-ADA1, and in this plasmid, we've sort of, we're encoding for tyrosin, which is sort of fused to a KNP1 restriction site in the plasmid. And this is sort of linked to an autotransporter called ADA1. Now, this autotransporter is what's going to be carrying it to the surface of the cell, and which is where the tyrosinase will be expressed. So basically, we are taking a promoter LAC-UV5. Now, it's important to know that this promoter is independent of any sort of induction or activation. And so essentially, as long as, there's a part of the cell surface where the tyrosin isn't there, this is going to be expressed. So then we're going to be taking MYC and His tags on either side of the tyrosinase gene, and we're going to be linking this to the ADA1, which as we mentioned before, is the autotransporter. So essentially, what happens is ADA-C takes it straight to the outer membrane, where this sort of linker is sort of attaching itself with the MYC antibody and the tyrosinase and the His tag as well. So,
Yeah, so this graph basically shows the results of what we've been doing so far. So the green squares here represent the biomass, the blue represent melanin production, and the red diamonds are showing the tyrosinase production on the cell surface. So one of our main goals for the product cell surface was that it would reduce cytotoxicity of the cell. And we can see that here. So even as we keep producing tyrosinase and melanin production keeps increasing, cell is still produced the same amount, which shows there's no effect with the metabolic systems which are already present in E. coli. And another graph showing basically the effect of selenomelanin with respect to pheomelanin and eumelanin. So here we can see that as the X-ray dosage increases, selenomelanin still has, so the cells which express selenomelanin still have close range at the exact same place, whereas the ones which express pheomelanin and eumelanin keep varying with the radiation, which is not ideal. So this shows that selenomelanin is an effective method to combat radiation. So yeah, so as I mentioned before, so we aim to produce biofilms with our E. coli, and we aim to create like an inner layer between two layers of the spacesuit, which can cover it and create like an effective method to absorb radiation. And on the larger scale, like much larger scale, we can even use this to coat like the inside of a space shuttle or rocket, since the astronauts are still experiencing high amounts of radiation even during space travel. So another thing we thought we could do is extract melanin from the cells and use it to manufacture like various other products. For example, the melanin-based sunscreen, which would be way more effective than our regular sunscreens used. Some challenges.
There's a lot of good that it could do, but before we get that, there are certain things that we need to consider. And the main is that, there's like main selenomelanin. We're going to need to have selenocysteine present as a substrate in the environment. That's be like sort of constantly, like we need to be constantly manufactured separately, and then we can't even able to like make selenocysteine in bacterial cells as such, like as just one amino acid. And so this sort of production of selenium also needs to be amplified, like actually make it work in the applications that we mentioned before, like the sort of having it as like a biofilm in rockets as well as spacesuits. But yeah, so it's just not enough right now, but there are tremendous applications.
Okay, thank you. Well, thank you. This was a very nice idea. And well, questions or comments? No. Okay. Well, there are various. Yeah. Yeah.
So maybe not produce. Okay, so it's produced on the surface, and then so in terms of enzyme, take the two substrates, form the selenomelanin, and then the selenomelanin release. So your biofilm that works in the space, the biofilm will have parasites, the bacteria, and then what cysteine is the medium. Yes, so cysteine and parasite is going to be in the medium, and like it's going to sort of release a lot of melanin in the surrounding environment. That's the melanin in that surrounding environment. The cysteine. So there's a problem because the cysteine molecules in general don't actually pass the cell without the help of some sort of transport. So we're not too worried about this cysteine itself passing. But again, selenocysteine is used even in bacterial cells. It's used as a natural inversion. In a small quantity, sure, but yeah, most of it won't actually go because so this sulfur and in our case the selenium and actually. The enzyme that is using translation, this is the natural enzyme for conversion. So the thing is, how is it? How do you like? Is it optimized enough to make melanin out of cysteine? Like, isn't the enzyme optimized to make melanin out of cysteine? So okay, right, but so the reaction that's actually happening to convert it to melanin does not really have any sort of binding with the sulfur of the cysteine in general. Wait, I'll just go to the. Is that sulfur is not
shown as the exact same. So if you use the enzyme method, they have shown that.
So again. Wouldn't necessarily interact with any of the other molecules. Like when we're making right, so you can see that it's super sort of it's not interacting with the direct aromatic chains on the field we are producing.
Any regulatory or structure that
Say that it doesn't have any effect at all, but we don't think it would be bigger.
Yeah, so like that was one of our challenges, and they only put small one.
Yeah.
Right, so idea was sort of like the biofilm here, and then the melanin produced acts in front of the biofilm, which is actually what the like right, right.
Okay, I have another suggestion. So what if you engineer the whole thing in a cellulose producing bacterium? Because at the end, what you may have is to form a biofilm. The biofilm gets well matrixed because of the cellulose and everything. And then you treat that with the procedures that we discussed this morning. And then at the end, you get a textile material that can be used to make some space suits rather than having a cutter, right? Because at the end, what makes the big difference is the melanin itself. I mean, whether or not this is together with a live thing doesn't make any difference, right? So how about it? It could be interesting, right? Right. Okay, very good. So let's write down the ideas and patent them immediately, right? No, but this is very good. Well, I think it has been very nice presentations by all of you. Yeah, thank you very much. And yeah, well, obviously, to make these projects a reality, you have to go through the nitty-gritty, to the details and everything. But in principle, I mean, what you have heard today tells you that you can just imagine different possibilities, and also that when you share this idea with the others, then you may even further refine the possibilities and come up with more interesting projects. Yeah, absolutely. Okay, great. So I think that we'll have more projects to discuss, and then we are coming to the final stage of the whole thing. I don't know whether I don't want to record that. Okay. Yeah, just stop. So everything that we say will be kept among ourselves, right? So we can stop that. Yeah. Okay. And okay. Well, it can be Zoom, but I don't want to be recorded. The what? Only Zoom, but not record. Okay. Okay. I mean, it's not that we're going to discuss state secrets, but I think that is good that we. I'm going to see your faces now.